So with that, I'll turn it over to the next session moderators. Thank you.
So the next session is a technology session on omics, finding a needle in a haystack, and I'm going to turn it over to our session chairs, Whitney Bessie and Brittany Lassaigne. Thank you.
Hi. Thanks everyone for being here. We'll jump right in since we're a little bit behind. Please put your questions and comments in the chat and we'll hold them all for the panel discussion at the end. Our first speaker today is Dr. Benjamin Humphreys. He received his AB from Harvard College and his MD and PhD from Case Western as part of the medical scientist training program. He trained in internal medicine at Massachusetts General Hospital and in nephrology at Brigham and Women's Hospital and Massachusetts General Hospital. He's chief of the Division of Nephrology and the Joseph Friedman Professor of Renal Diseases in Medicine at Washington University in St. Louis, and he also currently serves as president-elect for the American Society of Clinical Investigation. In 2021, he joined the board of scientific counselors for NIDDK, and as an internationally recognized investigator in kidney fibrosis, organoids, and regenerative medicine. His current efforts focus on single-cell multi-omics and spatial transcriptomics as approaches to understand and subphenotype human kidney disease. He's going to talk to us this afternoon about defining cellular complexity in human ADPKD by multimodal single-cell analysis. Dr. Humphreys?
Thanks so much, Dr. Lassaigne, for that really nice introduction. And I'm pleased to be joining you all today. I'm impressed by how big of a crowd you are. I wasn't expecting almost 200 people. And so it's a real privilege to be here. These are my disclosures. One of them is relevant, and that is with Chinook Therapeutics, who funded most of what I'm going to show you today. These are the brief stories that I'm going to tell you today, really a multimodal data set that we've generated on ADPKD kidneys, talking about proximal tubule and principal cell states, some validation of gene regulatory network activity, and then future work in spatial transcriptomics. And as a very brief intro to single-cell multi-omics, you will all remember from your molecular biology that our chromosomes and DNA are wound around histones, and most of the genome is tightly packed and inaccessible to both transcription factors and RNA polymerase, the transcription machinery. But pioneering transcription factors can cause loosening or relaxing or unwinding of the DNA around histones, allowing these transcription factors to bind to cis-regulatory elements and ultimately drive transcription. And so the data that I'll be showing you today measures both RNA at a single cell level, but also chromatin accessibility. And that's what I'm depicting here with this enhancer and then the promoter in the gene body. And because not everybody works with ATAC-seq, and that's the assays for transposase accessible chromatin, it's a pretty nifty technology. And so I just thought I would spend 30 seconds telling you how it actually works. And so it's dependent on this enzyme called TN5. And this is a hyperactive transposase. Transposases bind with very high avidity to DNA, and then they insert DNA stretches. And this mutated hyperactive transposase can be preloaded with DNA so that when it comes across chromatin that is accessible, it will bind, and then it will insert these oligos, which are essentially PCR handles. There's one in red and one in blue. And when two transposition events occur in one stretch of unwound DNA, you have a red and a blue sort of complementary PCR primers that will then amplify the intervening genomic DNA. Now, for the aficionados, you'll recognize that it's just a chance whether you have a red and a blue across from each other. You could get two reds, and that won't be that will not be amplified. Same with two blues. In any case, we are able to preload the transposase, and that's what allows us to map back by genome sequencing the library back to where the chromatin was accessible in the genome. And then the workflow is quite standard that I think most people actually are familiar with nowadays. We use the 10x platform predominantly. For multi-ohm, you do a transposition in bulk, and then you generate libraries, and you either run them as an RNA or ATAC, and then do a combined analysis. And so this is a collaboration, and I really have to thank the PKD RRC for this. And I hope that my talk illustrates the power of the core for bringing people like me, who, I think everyone would agree is not a PKD researcher, into the field, into doing research on PKD and PKD samples. And so we obtained these 8 samples through a collaboration with the core, with Terry, Owen, Stephen, and Paul when he was still at Maryland. These are pictures of the 8 kidneys. These were snap frozen, and we're able to generate nuclear suspensions from frozen materials quite routinely. And we subjected them to RNA, single-cell RNA, and single-cell ATAC-seq. And so again, a big thank you to the PKD RRC, who really made these studies possible. Here is the clustering, the UMAP clustering, if you will, from the RNA data. And we used, I think it was N=7 healthy kidneys. Now, these were not CKD kidneys. They're not, end-stage ESRD kidneys, and that's a caveat to this data, right? Because the only human PKD samples we can get are end-stage. And so there's a question about what the appropriate control is. Well, in this case, we used healthy human kidney as the control. We obtained 62,000 80 PKD nuclei, 40,000 control. We've co-projected them here, and you can see we get all of the usual suspects. We get fibroblasts, a bunch of leukocytes, lots of proximal tubule. And if you look over here on the right, we've shown, in orange, the cells that came from 80 PKD, and the teal is control. And there are some clusters where there's real overrepresentation of 80 PKD, and these probably won't surprise you. Lots of fibroblasts come from PKD. Most of the leukocytes come from PKD, and then there's contributions from all of these epithelial clusters as expected. A point I want to make, showing the data quality metrics here, is that it is actually kind of challenging working with these PKD samples. And so here we're plotting the number of genes detected per nucleus. So, we generally get 2,500 genes per nucleus in healthy samples, or the number of unique molecular identifiers, which is typically even higher. That's the number of transcripts. On the right, you can see our 5 controls, and these are by and large higher quality data sets than the 8 PKD, and you can see there's variability. And I think this just really reflects the fact that, number one, this is very fibrotic tissue, and so generating a nuclear suspension without destroying some of the nuclei and or damaging the RNA is actually hard. And also they've been frozen for years in most cases. Nevertheless, we're able to still do stuff with the data. And ATAC-seq, by contrast, we're measuring DNA that is much less susceptible to degradation, and we find that we get outstanding quality ATAC-seq data even from very marginal samples from the standpoint of RNA. So here's the ATAC data. We've now integrated it with the RNA, and you can see, again, on the right, you can see which came from PKD, which from control, fibroblasts and leukocytes, again, predominantly PKD. Now, if you look at the connecting segment and principal cells over here, or the thick ascending limb, or the proximal tubule, I just want to point out, there's this main cluster, which is the control, but then off to the side are the PKD cells. And what that means is that these proximal tubule cells from the PKD samples, they're still proximal tubule, but they're not the same as the healthy. And the same with TAL and connecting segment and principal cells. And that's a point that I'm going to make a little later transcriptionally, is it turns out there really are no normal cells in an end-stage PKD kidney. Here are some examples of different anchor genes in terms of their ATAC counts. You can see these are presumably all the transcriptional start sites for these various genes across clusters. This is kind of a busy slide, and I just want to make a few very general points. We are looking at the control kidneys on the left, the PKD on the right, and the different cell types on the x-axis. And the y is the gene set enrichment. And if you look up here in the upper left, there are low scores for all of these terms over here. What are these terms? Well, it turns out these are mostly inflammatory, like TNF alpha, IL-6, interferon, apoptosis complement, KRAS, EMT, TGF beta. And these terms are all high in PKD, and especially high in endothelial cells, fibroblasts, and leukocytes. Whereas on the bottom left, you can see kind of globally these terms which are high in normal and low in PKD. These are things, these are typically sort of peroxisome, fatty acid metabolism, oxidative phosphorylation. These are sort of energy terms that are down regulated. Now, back to this point of, are there any proximal tubule cells in normal proximal tubule cells in these PKD kidneys? We had previously done a multi-omic analysis of a mouse ischemia reperfusion and healthy human kidney. And just to make a long story short, is we were able to define a proximal tubule cell state that we called failed repair proximal tubule, and others have called this maladaptive. And it's characterized by expression of VCAM1. It's highly pro-inflammatory, pro-fibrotic. We think that it may be driving the AKI to CKD transition. Surprisingly, we can also find a subpopulation of these cells hiding out here in red in apparently healthy human kidney, and they accumulate with aging. Point being, this is a pro-inflammatory, not a normal proximal tubule cell state. And when we look at the proximal tubules from PKD, and I should say, by a sort of stroke of happenstance, all of this data that I'm showing you here is now in press as of this morning. We just got the announcement from Nature Communications this morning, so very, I think, auspicious and appropriate, since it's the PKD symposium. In any case, when we look at the proximal tubule lineage, and now we are just taking the 80 PKD proximal tubule cells here in blue, and we layer on top of them failed repair proximal tubule from healthy kidney, we see there's a good overlap. And what I'm not showing you is that if I overlapped healthy proximal tubule, I probably should have included this slide, with all proximal tubule from PKD, they don't overlap. They sort of connect down at the very bottom. But that really suggests that there are no true healthy proximal tubules in a PKD kidney. We can see 4 or 5, probably 4 different subclusters here. Number 1 has the highest expression of LRP and cubulin, so it's probably the least dedifferentiated. And then these other ones have various genes, whether they're FOS, immediate early gene. Prickle is in the Wnt pathway, TGF beta, KIM1. But we were quite interested by a gene that turned up. I think it's going to be in the next slide. Excuse me, just to finish this point, the majority of proximal tubule in ADPKD is altered. And so here we're looking at the different proximal tubule subsets from ADPKD 1, 2, 3, and 4. They are most similar by Pearson to this failed repair subcluster in healthy. And they're really not very similar to healthy S1, S2, or S3. And we can see that when we look at the interstitial space between like big cysts, where you can see non-dilated tubules that express cubulin, so they're proximal, but they also co-express VCAM1, which is a failed repair marker. And here's a tubule that's all VCAM1 positive with just a few cubulin, so it's probably quite dedifferentiated. There are a couple tubules here and there which don't express VCAM1, but here it's VCAM1 with LRP2, same story, really These tubules are not healthy. They seem to have this pro-inflammatory phenotype, even though they're not cystic. You can see these cysts off of the dark space on the side. There are similarities and differences. This is kind of getting into the weeds, but here's 1, 2, 3, and 4. 1, they all have downregulated oxidative phosphorylation, but 2 is the least downregulated. 3 and 4 seem to be the most inflammatory. We can see different cell signaling pathways that are upregulating these subsets: NF-kappa B, TNF-alpha, EGFR, JAK-STAT, and so on and so forth. But I think, perhaps, more interestingly or equally interestingly to this crowd, the collecting duct cell types, the connecting segment and the principal cells are also very altered. And here's what I was talking about. Teal is healthy connecting segment and principal cells, and orange is ADPKD. There's very little overlap. And so, and again, we can subcluster all of these principal cell. We call them CTC just to not confuse readers with like polycystin 1, PC1, PC2. But anyway, there's several of these different principal cell subclusters that are characterized by downregulation of markers of differentiation, like aquaporin 2 is downregulated, and then upregulation of these various other markers. And I want to focus on GPRC5A. This is an orphan G protein coupled receptor. You can see its expression is mostly in the principal cell clusters 1 and 2. It is this red, it's an apical receptor. Here we're co-staining for CDH1 and GPRC5A on this big cyst, and it's absent from control. And it's a kind of an interesting gene. It's mostly described in cancer, where hypoxia upregulates this receptor, where it's hypothesized to be a secondary microenvironmental sensor, but its ligand is unknown, and it promotes fitness of these cancer cells. And that kind of made sense to us, since these epithelial cyst lining cells must be hypoxic. But then I found, and I only just found this actually in preparing for this talk, there's this bioRxiv paper from over 2 years ago that still has not been published. So take that with a grain of salt, where they have described this same receptor in PKD signaling, where they conclude that it's interacting with PKD and regulating trans-Golgi protein trafficking to the basolateral membrane, and that it senses protein overload in the trans-Golgi compartment. So I don't know what to make of that. Maybe some of you do, but it's kind of an intriguing connection to the cell biology here. So just to illustrate finally the kinds of things you can do with these multi-omic data sets, we can generate these cis coaccessibility networks, which is a way of predicting cis regulatory element regulation of gene expression. And so here we've got our friend GPRC5A. Here's the transcriptional start site. And this cis coaccessibility network basically is asking, well, when this piece of the genome is open or accessible, what other parts of the genome are also open? So it's a statistical calculation. And basically the network here shows that this upstream 5' part of the genome is a potential enhancer or a potential cis regulatory element. It's also interacting with some other genes and candidate cis regulatory elements. Well, we can test the prediction that this enhancer is driving GPRC5A activity using a dCas9. So we can send a Cas9 fused to a Crab domain, which closes chromatin. And we can send it to this locus with guide RNAs. And you can see that when we do that, first to the promoter, which is an internal control, we of course turn off gene expression, gotcha. But that we can turn off 60% of gene expression when we target the enhancer. We can also look at the kinds of transcription factor binding motifs, retinoic acid and CREB1, and we actually showed in the principal cell line that activation of these signaling pathways can also drive up expression of GPRC5A. So I just want to say that all of this data is on KIT. And let's see, when we get to the chat, I will put the password for the Muto et al. Data set, so you can check out the PKD. I'll put that in the chat as soon as I'm able, and you can look up your favorite gene. So I just want to finish with the final minute to tell you where we'd like to take this. I think we're really investing a lot of effort into spatial transcriptomics. Here's Visium, and here is a Cartana, which is a sort of similar technology, in situ sequencing. And just to point out that there really are rapid gains being made in resolution of spatial transcriptomics. I probably don't have time actually to go through, but if there are questions, I'd be happy to. It's an in situ sequencing probe-based technology. And when we compare, Visium, which is very robust, I mean, we actually like it. It's genome deep, even though it's quite low resolution. You get really much better resolution with this kind of Cartana approach. And then with a tremendous amount of work, we were able to figure out ways to segment cells. And you can see here we're looking at podocytes. Each dot is an mRNA molecule, a single molecule of podocalyxin, and we can now begin to reconstruct glomeruli in this spatial map. And here's a sort of a higher resolution image of a whole kidney with these 6 genes according to this Cartana. And so the question is really, we'd love to apply this to PKD models. I will stop there, and I really want to recognize Yoshi, who's the first author on the manuscript that I presented to you. Also, Erin, who got her Ph.D. From the University of Maryland, and we're thrilled to have her, and especially to thank all of the members of the PKD RRC who made all of this work possible. Thanks so much.
All right, thank you so much, Dr. Humphreys. That was great and really exciting. Looking forward in the field.
Okay, I just put the username and the password in the chat if anybody wants to look up their favorite genes.
Great, thank you. The way this section is structured, we're going to go right on to our second speaker, and then us chairs will try to field the questions as best we can to lead to a lively discussion with the panel after the speaker. So let me introduce Simone Sanna-Cherchi. He's an associate professor at Columbia University and a member of the Institute for Genomic Medicine at Columbia. He's a nephrologist by training and also a human geneticist, and his lab has mainly focused on the genetic basis for congenital anomalies of the kidney and urinary tract, as well as idiopathic nephrotic syndrome. His lab continues to work on those, and he's made major contributions to the field and prominent journals. So we're really glad to have you, Simone. Take it away.
Thank you very much for inviting me to this beautiful meeting that, unfortunately, as you can see by my pajama, I'm on clinical service, so I'm not able to follow the entire agenda, but I'm seeing like a tremendous work, and it's actually humbling to be here. Let me try to share my slides so I can go through. Okay. Everything seems to work okay, right?
Looks good. Thank you.
Okay, so I really don't work on PKD. So hopefully today, since it's a technology session, I can show you and use examples from my personal interest to show certain type of approaches that are now possible, and we need probably to fine tune a little bit, and what could be applied to PKD research. So my talk is reconciling sample size and accuracy in genetic studies for kidney disease. And I have, if I can move to slides, minimum disclosure to make, really no real conflict of interest. So I'm gonna use as a phenotype of interest for the talk of today, idiopathic nephrotic syndrome, that, as you can see from the photo here, is a primary renal disease that is defined by very heavy proteinuria, so loss of protein in the urine with swelling, edema, and low albumin that comes with a lot of complications. So the two main histopathological categories of idiopathic nephrotic syndrome are minimal change disease and focal segmental glomerulosclerosis, or FSGS, that they show some commonality and differences, but they have as an ultimate target cell the podocyte. So there's particularly for FSGS, is progressive, is a localized scar of a subset of the glomeruli. It's very rare, but is a very prevalent cause of nephrotic syndrome in both children and adults, and there's a very high progression to end-stage kidney disease, as well as a high frequency of recurrence post-transplantation, of which we don't know almost anything in terms of genetics. So needless to say that we need better understanding, and for the better understanding, in my perspective, is genetics. So I'll go through and so tell you how we're going to try to tackle in a collaborative fashion issues about genetics of FSGS. So this comes from a coalition from 4 PIs, including myself, Martin Pollack, you see him here on the top, Matt Sampson, and Frida Hildebrandt, in which we put together this coalition to try to accelerate the genomic discoveries on FSGS and nephrotic syndrome by mostly by aggregating all data and samples that we accrued over decades of tedious recruitment of patients across all the continents in order to conduct like large-scale genomic studies. So as soon as we put this together, we actually had other individuals joining this coalition, Rashid Badegizin at Duke and Moin Salim, as well as the QGN consortium. So now the idea is really to combine all these cases together, leverage on genomic sequencing and genome by genotyping in order to solve the etiology of disease. But there are multiple caveats to that. So I'm fortunate to be sitting at Columbia, where at the Institute of Genomic Medicine that was founded by David Gornstein, now the interim director is Al-Gharavi. We basically have an honor system based by which every investigator that is interested in genomics dumps his exomes and genome in a common warehouse that we now adapts to about 120,000 exomes and about 20,000 genomes that they all pass through similar QCs for data harmonization. Is an honor-based data sharing type of process in order to foster collaborative gene discovery. So that's where all the genomic data for this coalition sits for nephrotic syndrome. And we are now in phase 1, where we actually coalesced about 6,700 in reality. The latest cohort is still pending, but we are getting there. So this is phase 1. We have about 6,500, 6,400 individual with nephrotic syndrome with the genomic sequencing, either exome sequencing or full genome sequencing, and accompanying DNA microarray in order to extract all sorts of the genomic variation that we are interested in testing. And I'll use this cohort and parts and bits of these cohorts to show you some of the studies that we are conducting and what are the pros and cons of putting together these type of studies. So one, just briefly, is a clinical genetic study on nephrotic syndrome. So here we look at 2,000 cases from mostly from the Columbia cohort and across ancestry, across age of onset, and across sensitivity to immunosuppressive therapy. So sorry, I just realized that this table got cut on the right, so not a good start. So they're just showing that we're trying to look at broadly and the entire lifespan spectrum as well as a phenotypic category. So first thing that comes, and this is going to be important for then the next studies. So this is the workflow. Ignore the part on the right side. This is a second part of this manuscript that we're putting together, but every single one of these 2,000 samples has been subjected to exome sequencing and genome by genotyping by Illumina array, and we then develop a diagnostic testing protocol. So first we test a panel of genes from the exomes that when mutated cause FSGS or related phenotypes, and we diagnose about 11% of the cases. We then look at APOL1, other 5% of the cases, and then all the ones that are negative for these genes, we tested for the remaining nephropathy associated Mendelian genes that are 700, and we find an additional small percentage of these ones. So basically, if you take patients, they come into your clinic with nephrotic syndrome, you'll make a diagnosis just, randomly on 10% of the cases, 5%, but, with a lot of caveat, they have APOL1 high risk depending on your ancestry background of your population, and a very small proportion they will have mutation in genes that are basically bystander. So this is a gross approximation, and by just modeling 2 parameters, so age of onset and response to therapy, you will see that the distribution of what is known already now, it varies significantly. And this has implication then for discovery. So when you look at the age of onset, if you look at the over time, less than 5, less than 15, less than 21, and so on in age, so the percentage of the diagnostic rate drops, so from 20% to the baseline that we said of about 10% in total, but it changes significantly over time for the forms that are bad, so steroid resistant, in which, in 23% of the cases we make a diagnosis in age 5 or less, and in 15% at age 50 or less or more, sorry, while for the steroid sensitive, you have a small percentage, very, very few that they have a diagnostic variant in the early ages, and this increases a little bit over time. Actually, by segregating only on steroid sensitivity, you see that basically all the one, if you have a mutation in a Mendelian gene, so this is what is basically known in the literature already. If you have a mutation in non-Mendelian genes, so you are steroid resistant, while there starts to be a background frequency of these mutations while you start increasing with age, and this reflects our increasing complexity of these phenotypes once you move from the pediatric highly genetic forms of disease to the later stage. And that's we'll come back to these concepts when we discuss discovery. So really, we are interested in using our aggregated data for gene discovery, and this is one of the examples that is published. So how we can leverage big data to lead to discovery. So this study comes from basically an anecdotal finding. So Patricia Wang at UCLA identified a kid that at 2 years of age developed seizure as a epileptic encephalopathy with developmental delay, and that she sees him at age 4 for heavy proteinuria and rapid progression of kidney disease. The biopsy shows FSGS, and that's the biopsy. So she runs a clinical exome and trio-based, and the result is a truncating mutation in this gene called TRIM8 that was associated in some case report with epilepsy. So now we come from another doctor. Is FSGS caused also by this mutation, or is a unlucky kid that has epilepsy caused by TRIM8 and FSGS? So we went on and looked at our cohort. So we screened exome sequencing for 2,500 pediatric cases with focal segmental sclerosis and compared with 48,000 internal controls at the Institute of Genomic Medicine. Everybody was sequenced with all exome sequencing or genome sequencing, and we identified additional 8 individuals with truncating mutation in our cases and 0 in control. So just by association, this leads to a p-value for association of 10 to the minus 11, so establishing basically a causal role. We look at the reverse phenotyping. Every single individual in this cohort had epilepsy or developmental delay. So linking together the 2 phenotypes. So now from another doc, we get to the fully recognizable syndrome. And when we look at the parents, every individual that had parents available, the mutation was de novo. So by leveraging on this large cohort, we basically establish causation, but we can also establish it the other way. So we look at 9,000 individuals with epilepsy, again, all exome sequencing on all of them, and we identified 3 additional cases with truncating mutations in TRIM8. And when we did the reverse phenotyping, all of them, they had FSGS or steroid-resistant nephrotic syndrome. So again, this is a proof how you can actually find the needle in a haystack by leveraging on this large cohort when you have a very defined gene and mechanism and genetic mechanism underlying, like de novo mutation. But we are also interested in, assaying the entire genome to look for novel genes. And that's where things they get a little bit more complicated. So the idea is that we can test the case controls, our 6,000, 5,000 individuals that we have against the 40,000 controls and see what's the distribution of rare variants. Here, the problem has always been controlling for all the confounders and bias that can happen when you aggregate data that they come from different parts of the world and sequence in different ways, different exome capture, genome sequencing, exome sequencing, ancestries, and so on. So Gundula Povisil was incredibly talented postdoc who still work with us, although she's now in the biotech. She developed a very rigorous mechanism, workflow for mitigating all this bias that is based, yes, on standard QCs, analysis, and so on, but she basically lifted methods from the single cell RNA sequencing work that you've seen before beautifully from Ben. And basically she said, if we can really have an incredible precision in discriminating cell types, maybe by using the same Louvain clustering methods that are used in single cell analysis, we can actually identify in a much more precise fashion individuals in the genomic space. And that's what she's done. And this allows basically perfect mitigation of all the confounders from different platforms and sequencing, as well as ancestry, and then allowing us to perform correct exome-wide case control rare variant studies. And that's what we did. So this is an interim analysis. The analysis of the complete data set that I mentioned before is ongoing as we speak. So this is about 4,000 cases and 16,000 controls with nephrotic syndrome, with all the cohorts provided by us. And we've done this test, and as you can see, it seems to work pretty well. So this is a Manhattan plot, but where every single dot is not a SNP, is actually a gene. And we test the enrichment, so the burden of rare variants of using certain parameters in cases versus control. So the first things that you see all the classic and expected culprits. So this tells us that this method works really well. So 10 out of 12 of the top rank genes are known FSGS genes. Some are novel candidates. And we're basically reaching the sample size for discovery. And if you really want to zoom in, that's a QQ plot. You can see the lift is, the magnitude of the lift from the null distribution is impressive. And interestingly enough, it works even for recessive model that traditionally they've been based on homozygosity mapping and use of very unique families with multiple individuals that are affected in single generation. But here we can actually leverage recessive models in a case control fashion. And again, you can see that works. It works also for recessive genes. I don't think it's 8 of the 12 that are ranked, but maybe yes. But again, you see a massive lift for all the known genes, MPHS1, so this nephrin, podosin, laminin beta-2, SMARCAL, and then a bunch of novel candidate genes that we would have not suspected by manual curations and so on. So it's really powerful as a way when you have a very discrete and specified hypothesis. But these systems, they also allow us to start looking at different genetic space. So you can start looking at these rare variants that escape the analysis in the, let's say, in pedigree-based, classic family-based studies, but they also escape because they're a little bit more common. They display a very incomplete penetrance, but they also escape GWAS because they're too rare actually to be picked up by linkage equilibrium in the common variant analysis. And they work also in these approaches, they work in this genomic space, but you can see something interesting. So you can see that in dominant models, they start bleeding in alleles for recessive genes. So this tells you that you can start looking at these this specific genomic space, a range of allelic frequency and effect size, but you can't assign any more mode of inheritance. But you're finally looking at a region that we were always blind to. So I'll get quickly on the rare variant association analysis and considerations. So sample size matter, but again, here we need a very specific hypothesis behind it. So here it's mostly a study that is driven by cases that are severe and they have no response to therapy. And no matter how large the sample size is, the study, they cannot disprove a gene. So you see over and over small size sequencing studies in which you sequence 200-300 samples, they don't find a mutation in certain genes and they say this is not, and they question the validity. In our study, by studying 4,000 samples, we don't pick up any signal for, some of the major genes like alpha-actin 4 discovered by Martin Pollack, and we would be reasonable if we use our study to say that that's not an FSGS gene.
3 minutes.
Yeah, I'm getting there with the very last part. So we can establish the mode of inheritance, and we talked about it, and we finally can study what we were able to study with the classic Mendelian approaches, very powerful, pedigree-based. You find the rare alleles that they cause disease. GWAS, you can find variants that are associated, that they have a small to moderate effect size, but now we can really start looking at this genomic space that is probably more reflective of the non-Mendelian forms of disease. But we're also looking at the GWASs in our studies. And this again is an interim analysis about 4,000 cases and 40,000 controls. And I'm going to show you very briefly what happens when you start. You see GWASs that are run in massive sample size with very limited phenotypic discrimination, and that could be an issue. So if we run 4,000 cases of nephrotic syndrome versus 20,000 controls, that's what we get. We get across race ethnicity, giant peak for APOL1, big peak for HLA, nothing else, right? So we already know that that's not true. So you make an estimate of, your test statistics gives you an estimate of effect size for APOL1, but we know that this comes exclusively only from the individual of recent African ancestry. And the HLA effect size is probably not the one that is driven in this population that yet is all nephrotic syndrome and perhaps some new genes. So these include in reality adults, children across ancestry, irrespective of response to therapy. The idea is that, they share some commonalities, perhaps we can find something. But my argument in this case is that, yes, maximizing your sample size, but reducing it in order to increase accuracy is actually beneficial. And so if you look at, simple parameter, the phenotype. So if you reduce and you look only at pediatric steroid-sensitive nephrotic syndrome, everything is completely different. You drop 1,500 cases, but you have 10 loci that are of early genome-wide significance. APOL1 goes away, not because the individual of African ancestry, they go away, but because it's not biologically relevant to this subphenotype. And then you see the true effect size of HLA in this subset of population, just to show you what it was before the Manhattan plot, very different. But what if we add phenotype, but also biology? Let's look at the steroid-resistant nephrotic syndrome. Again, here is APOL1, just 800 cases with for a GWAS analysis, reasonable, you see APOL1. And so the most parsimonious explanation is the sample size is actually a problem, but here the problem is that you have a big component in this category of disease that they have Mendelian mutations. So, and this they can really mask signals that are of moderate to low effect size. So we are fortunate we have exome sequencing in these cases, so we can remove it. So we actually drop our sample size, and the Manhattan plot looks very different. And now we see loss that come up at genome-wide significance, including HLA. Now here that they have very large effect size for a GWAS type of approach, and they were completely masked. So by increasing accuracy using phenotypic data and genetic data that they're a proxy for biology, we can really start dissecting subphenotype of disease that like this type of FSGS that is more likely to be multi-drug resistant and recur after transplantation. So this, I think, brings to the only slide that I have on PKD and how I can envision a study that in which we can learn from what I'm doing now and that can be applied to PKD. So one important thing, and this is just one example. So the genetic background modifies the severity of PKD, both in the age of onset, initiation, and cystogenesis. And this is a beautiful paper which you show that if you put a single, the exact same mutation, different genetic background, you have a different age of onset for the cyst formation, the different progression. This has been done in multiple, this was just one of the examples. And this also fits in with what we see in PKD. Well, you guys all know much better than I do that the age of onset and the progression is very variable even within a family. So is there a question for identifying genetic modifiers? And to my knowledge, there's no study so far in humans that they unequivocally prove the existence of a genetic modifier. And perhaps the tendency is that by lumping all together PKDs in order to favor sample size, this might actually be counterproductive, because we already know that the different mutations, different genes, they have a different severity. So the actual variation is within each group. So perhaps stratifying correctly individuals and designing appropriately human genetic studies will allow us to use some of these tools that I showed you for identifying modifiers of age of onset, severity, progression of PKD. And that's just a thought. Also because I'm particularly interested on another disease that for which we're looking for modifiers, which is APOL1 associated FSGS, and we are encountering exactly one of these issues. So the modifiers for FSGS are going to be very, very different than the modifiers from other APOL1 associated kidney disease. So I'll conclude by saying the sample size matters, but phenotype matters. So we'll have really to find a way to mitigate how we define our needle in the haystack and how we define our haystack. So the haystack in the early days were the genes. So it was very difficult to sift to 20,000 genes, a million of variants. Hence, early geneticists, they wouldn't go for initiating any study without a clear phenotype. Now we have actually readily available in chip sequencing, so we can assess and sequence 1,000, if not millions of individuals, but then we go at the expenses of the very large phenotypic variability that can lead us to potential hurdles. And I'll finish by acknowledgement all the collaborators and my lab and the founders.
Great, thank you so much. So I'll welcome people to start putting questions into the chat. And thank you, Simone, for a great talk. That's exciting to be in the interest of gene discovery, to see the resources you have and how you're putting it together. Let's see. I think, why don't we introduce our panel? We have Luis Menendez from the NIH, Maria Irazabal from the Mayo Clinic, and Kurt Zimmerman from University of Oklahoma. And we'd love to get a lively discussion going regarding multi-omics technologies. We've just heard a lot about technical advances as well as collaborative efforts. And so does anyone want to get started with questions?
So I think we had one question in the chat that maybe we could use to kick things off. And I think, certainly, Ben, if you're with us, you're welcome to join us for the panel. And then Kurt and others may be able to weigh in on this as well. So the question was about these changes that are seen in end-stage PKD and if these are unique to proximal tubular cells or if we see these changes in other cell types as well. Can anyone on the panel comment on that?
Oh, wait, here I am. Okay,
There we go.
I was disabled from opening. So the question is whether these failed repair states that we found in the PKD kidneys are present in other.
Yeah, is this
diseases?
unique to those proximal tubular cells or do you see these in other cell types as well?
I don't think there's a straightforward answer to that question. I think it depends on if you're a lumper or a splitter. If you're a lumper, then definitely we see this kind of pro-inflammatory VCAM1 positive proximal tubule cell state in almost every data set that we analyze, including, surprisingly, backbench kidney live donor transplants. So those, of course, are considered to be the gold standard of the healthiest kidney you can get. And yet we still clearly see these failed repair cells in that healthy kidney that has just been, explanted, which is intriguing. If you're a splitter, though, you could see that we could identify 4 different flavors of that failed repair state in PKD. And so whether those are all present, I don't know. I would tend to think at least some of them must be unique to the cyst environment, which is so unique to PKD, or at least to cystic diseases. So it's a kind of a vague answer. It's sort of like, definitely we see it in other diseases, but I think some of these states are probably PKD specific.
Hey, Ben, I guess I'd like to ask a question regarding that as well. I think maybe what the question is getting at is, do you see failed repair if you'd give an injury to, say, a distal tubular cell?
Okay, great question. So we have a paper coming out on that in later in a month or two, where we did look at shared and distinct epithelial responses to, in this case, we looked at unilateral IRI and UUO. And it's like a Venn diagram. I mean, it's probably not surprising. Like, there are certain hypoxia pathways and DNA damage pathways and heat shock protein pathways that are common throughout the epithelial segments of the nephron. Especially the thick ascending limb is the most similar, which may reflect its kind of location adjacent to the S3 segment. But then there are, at the same time, very segment specific injury responses, which I think are quite interesting. We haven't really, we've defined them, but we haven't sort of delved into the cell biology. But the ones that are shared are largely around these damage response, like, as I said, hypoxia, heat shock protein, DNA damage.
So I guess one other question I had, so going back to your lumper and splitter kind of approach, so how much of that is driven by patient-specific samples? For example, you mentioned that you found these 4 different types in PKD. Are they present in all samples, or is that kind of unique? And how do you interpret all of that?
So I think one of the reasons that we and others have been able to define these maladaptive, or what we call failed repair states in the proximal tubule is simply because it's the most abundant cell type. And so your sensitivity to detect these cell states is directly proportional to the cluster size that you have. And so it's a tricky question to answer unless you normalize the size of the different populations. So what I can say is that we do see this failed repair signature. We've never not seen it, including even in the healthiest kidneys. We have done deconvolution of bulk RNA-seq human kidneys and concluded that that failed repair, that maladaptive signature increases with age, and it increases with any disease state that we've been able to find, like diabetes or glomerular disease. And so what we think is that these are bad actor cells, and that if we had a way to get rid of them, kind of like senolytic therapy, and these cells do share a kind of a SASP, a senescence-associated secretory phenotype-like signature, in the sense that they are secreting a lot of cytokines. They're not senescent, though, transcriptionally. So that's a kind of an interesting twist. But we do feel like if we could get rid of them, that you would rejuvenate the kidney and make it younger and healthier. Does that answer your question? I think I might have only answered part of it.
Yes, I got to answer something different, I guess.
Yeah.
So yeah. So I think the problem I have with some of these approaches, and I'm not sure it's a problem that can be easily solved or not, but it's just the fact that if you now have all these samples, so for example, you mentioned that the identification of these clusters is going to be highly dependent on the number of cells.
Mm-hmm.
So if you now have a sample that has one large cyst, so it's going to be predominantly that many more cysts of that cell type. So that's going to already introduce a bias. Then you have one patient which is going to have like a very fibrotic, so it's going to have like larger number of fibroblasts. So you're already going to be difficult to compare that. Then you have all these response cells. So you have like all these presumably normal, kind of at least previously normal proximal tubes that are being compressed, and now they are responding to the compression, and they have their phenotype. So this is going to be all a little bit unique to each sample. And then when you analyze, we sort of lose track of what is coming in aggregate and would be true no matter how you resample versus it's true. But if you remove like this patient here, then the signature would all be gone. And so I guess the question is, how do you foresee this moving forward in terms of applying this to larger samples or identifying more sensitive markers?
Yeah, so I mean, there's a lot there, and so I'll just try to be brief. When we looked at the UMAPs of all 8 PKD samples, there was no one patient that was driving a cluster. Every subcluster was contributed to by all 8 patients. So I don't think your last point is a huge one, at least if you have a sufficient N and your samples are sort of similar. I think the question, the point about, cyst lining cells versus compressed, non-cystic tubula is a really good one. And that was one of the biggest surprises in this analysis, was that we couldn't find any normal. We were going to use the quote normal proximal tubules as the control, as the internal control, but we couldn't because they were so abnormal. And really one of our reviewers was really hung up on this whole issue and thought that there was a systemic problem in our data set. And so we had to bend over backwards to show that there really weren't any normal proximal tubules in this kidney. I think future technologies will help address some of those concerns and points that you make. I think spatial technologies, right? I mean, that's obviously what is needed here, so that you can tell, well, what are the cyst lining cells, and then what are the tubules that are adjacent, and what's different in terms of their gene expression. Another thing that's interesting that we are pursuing is that it turns out that single nucleus ATAC-seq data, and Simone may comment on this, is actually like low-level whole genome sequencing. Like, we can detect with good sensitivity in a reasonable sized cluster somatic single nucleotide variants, and we can call them with high confidence. Now, we tried to distinguish cyst epithelia from like failed repair epithelia using that strategy, and we failed because I don't think we have a deep enough sequencing. We didn't design the study to do that. But you can imagine the day where you'd be combining whole genome sequencing or single variant calling and looking for mutations in PC1, and then second hits and third hits, and looking for clonality in the cyst lining epithelia. And so that's something that we're very interested in and are developing pipelines to pursue. And so I think that the future technologies will help address some of the limitations of current technologies.
So Ben, I have a question, I guess that's kind of addressed, is you mentioned briefly, is you don't see any normal tubular epitheliums in your end stage kidney. Do you think that would be the case if you looked at earlier stages as well? So like maybe profiling, say, urinary samples to look and see if you get any normal ones popping out and projecting that back onto your end stage signatures?
Yeah, thanks, Kurt. I mean, it was a little frustrating. Like when we had designed the experiment, what we thought was we would be able to distinguish like early small cysts from like late, and then we would focus on the early small cysts and be closer to like the cyst initiating of signaling rather than all these adaptive changes. And we clearly couldn't distinguish, small, large cysts at all. And so your point about trying to get tissue from earlier in the disease is a good one. I agree that for sure urine would be a great source. That's not something we've pursued a lot, not for any particular reason. I am happy to say, and thank you again to the PKD RRC, we are just going to be funded for a pilot study to do spatial transcriptomics on Terry's inducible mouse model. So I think, we'll do a time course of early and mid and then not late, and I think the mouse models with these kinds of approaches, and I'm sure other people are doing that, are going to hopefully yield some of these much earlier proximal signaling events.
I just want to add something about the limitation of using late stage tissue samples for obvious reasons. But my thought that, and I am very skeptical of using late stage kidney samples on how much is coming truly from ADPKD and how much is coming from CKD and renal failure. So, but one thought could be of comparing it with other type of ESRD forms. And I was wondering whether if we could compare those, because I think one of the advantages of using the kidney tissue as opposed to using culture cells or even cells from the urine is that you allow these cells to be surrounded by the whole environment and the changes that CKD could potentially do to these cells, because the cells are not isolated. So I wonder if potentially we could use this technology and compare it to other forms of CKD, like for example, diabetic nephropathy, and study proximal tubules or collecting ducts from other forms of CKD, and even also interesting from other forms of polycystic kidney disease, like an end-stage kidney from ARPKD, and think that potentially we can identify signatures that could be related to CKD, but also potential signatures that could be related to the cystic phenotype, or that there might be signatures that could be related to ADPKD or ARPKD. Any thoughts about that?
Yeah, totally. And I mean, that's exactly sort of along the lines of what we're thinking as a consequence of the results of this study. And I mentioned it at the beginning of my talk, is that, you can really argue about whether we used the correct control or not. So in the past, when we had the option to collect human end-stage kidney disease samples, that were being explanted, we were like, well, who wants that? But we've started collecting them, and we've got several, and I think we're doing that for exactly the reason that you say, which is it seems plausible that if we then generated a multi-omic data set of these end-stage kidney disease from non-cystic causes, that again, we can do this, co-project and then look at where the differences are amongst the proximal tubule, and you can see it, subtract out all of the failed repair hypoxic interstitial tubules, and then really focus your analysis on the gene changes or the cells that are specific to ADPKD, and extend backwards and along the whole CKD timeline. And for CKD, we're already doing all of that, and we've published some, and we're doing more. That's sort of a core interest of ours. But I agree. I think that's probably what we should have done, from the start. It's just that it felt, these studies are so expensive. It felt like wasting a ton of money to profile like an end stage kidney.
Simone, I
yeah.
really enjoyed your talk also about the reverse phenotyping and the epilepsy and TRIM8 story. I wondered if you could tell us, like, where else do you think that could be applied? Like, are there other studies that you're excited about with this reverse phenotyping?
Well, this is all in the effort of the phenotypic expansion, right? So we already have many genes. We have about 4,600 genes that they have, when mutated, they have an assigned phenotype, but we only are looking what you are looking, right? Neurologists, they look at the brain, we look at the kidneys and so on. So we see coming on from our studies that, you know, when someone is very sick and there's a major reason to be sick, that's what gets discovered, right? And then you go back, and so we have a few additional phenotypic expansions coming from the cardiovascular space. So children with congenital malformations that you know, the kidney phenotype was completely overlooked, and we went from using genomic studies combined with transcriptomic studies based on mouse models that we have in the lab for genes to actually reconcile syndromes in which the unique genomic lesion is a pleiotropic effect. So that's one thing that we're doing. We're seeing mostly for cardiac and neuro, but again, cardiac and neuro are two big specialties in which there's a lot of gene discovery. We are actually also collaborating with some providers of genetic testing that they run all these Mendelium type of test across the country, across the world, and they accrue the tens of thousands of individuals. So now we can actually test specifically for this phenotypic expansion. So
3 minutes.
yeah, one thing that I actually have a question for the group, because you know, as you know, I'm super far from PKD, but I was kind of surprised that I haven't really seen a human genetic study attempting even to find the modifier of disease severity. There's a lot of data in the mouse. In the humans, there's some sequencing additional sister-related genes and looking at the coexistence of rare mutations. But is there any ongoing effort, like NIH sponsored or whatever, to accrue the cohorts of genetically determined PKD1 and PKD2 in order to look at modifiers? Are there?
So I believe Peter Harris has done that type of analysis. I don't know if he's probably not able to unmute. I don't know if Maria, you're able to speak to that at all.
Yes, Peter Harris at Mayo is conducting study to identify different genetic modifiers in typical PKD populations. But Peter, if you are around, if you would like to say more about the study, that would be great. But definitely, we have seen a huge phenotypic variability within even families. So definitely, Peter Harris at Mayo is conducting a study to identify different genetic modifiers that could point out. And also, we are interested in environmental modifiers, and that's why we are complementing those studies with metabolomics analysis and trying to characterize the phenotypic variability that there are among these families. But there are, yes.
Yeah, I'm asking, is a tricky business, the one of the modifiers, brought from a methodological standpoint, but also patient selection and so on. And we are dealing it for APOL1 modifiers in our stuff. Okay, we are doing a GWAS of PKD1 patients, and they're working on this. That's interesting. Yeah, because to see what leads. And quick question to Ben. Are you paying the royalty to David Hasselhoff, or you're infringing copyright by using Kit in your website?
I think David would approve, don't you?
I think so.
And you realize that like half the people on the call probably don't even know what Knight Rider is.
I was a big fan when I was a kid.
So maybe we can just ask a question to Dr. Simone. So in your data set, so are you able to find like multi-allelic allelic diseases. So for example, if you just try to find phenotypes that are associated with 2 mutations that by themselves would not lead to disease, or is that something that would be
Biallelic. So that's a very good question. That's I think what some of these methods also can prelude to. So before testing digenic inheritance was really complicated. I think we are actually reaching a situation, especially when we have you can phase and you can look at trio-based studies. You can start testing, by fixing one gene and then looking at the coexistence with other. You can start testing digenic or multigenic inheritance. This is again a question of what's the sample size and the effect size that you need to discover those type of. So we are doing for some specific phenotypes that we think that despite the rarity and the likelihood for being highly genetic, yet we can't find a single smoking gun. So there must be such an example of digenic inheritance. Yeah. These are testable now. I don't know yet. I don't have the answer to see how for sure there will be example. I don't know if in our field.